I want to briefly go over some of the histology of the digestive system, since I think that is what most students struggle with in this particular um, chapter in their lab manual. So the first thing, of course, we're going to look at is the esophagus. So we've got our four tunics. We've got the mucosa, the submucosa, the muscularis externa, and the adventitia. It's not the serosa at this point. It's a more fibrous connective tissue called the adventitia because the esophagus is not within the abdominal cavity. So one of the things that makes the esophagus different from the rest of the GI tract is it has stratified squamous epithelia in its mucosa. Most of the rest of the GI tract has simple columnar. The submucosa is pretty standard. It's the areolar connective tissue. And then there's another modification in the muscularis externa. There's skeletal muscle at the superior end of the esophagus, so towards the head, and there's smooth muscle at the inferior end of the esophagus, so near where the esophagus joins with the stomach. We still have our two layers, our inner circular layer and our outer longitudinal layer, but it is the muscle type that differs, transitioning from skeletal muscle near the head to smooth muscle near the stomach end of the esophagus. And so if we kind of look at them both in cross-section, so here is a section cut transversely through the esophagus, and then in a longitudinal section. You can see the four layers. You can see here's the mucosa in the longitudinal section. Here is the mucosa in the circular section, the cross section. This would be the lumen, because here's the mucosa, here's the stratified squamous epithelium. So this is the lumen of the esophagus. That's what this is right here, the inside portion. So this is where food is traveling down. It's inside this lumen of this hollow organ. You can see the lamina propria as part of the mucosa in both regions here, both sections, and the little bit of muscularis mucosa that is also part of the mucosa. And again, you can see it on both of the cross-section and the longitudinal section. Just below the mucosa is our submucosa. So here it is in a longitudinal section, and here it is in a cross-section. So all of this here is part of the submucosa. And then below the submucosa is our muscularis externa. So here is our muscularis externa. Here is all this. All this is muscle, 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 muscle. We've got the inner circular layer. So that's what this would be here. And then the outer longitudinal layer. So that's this here. And then below the muscularis externa, we have our fibrous connective tissue, the adventitia. If we look at the gastroesophageal junction, this is where the esophagus meets the stomach. So the junction is actually right here. It's that very abrupt stop from the stratified squamous epithelia of the esophagus to the simple columnar epithelium of the stomach. And if you look at it in a closer view, this is a higher objective than this particular picture here. You can see here is our stratified squamous epithelium of the esophagus, and you can see the columnar cells of the gastric epithelium, the simple columnar. So there's one cell, there's another cell, there's another cell, just one single, simple, tall columnar cells versus the many, many, many layers of the stratified squamous of the epithelium. So you should be able to differentiate between these two parts of the GI tract if you were to see them on a lab practical. So food enters the GI tract through the mouth and the oral cavity, travels down the esophagus, and then enters the stomach. So we still have our four tunics, the mucosa, the submucosa, the muscularis externa, and now we have our serosa, so areolar connective tissue. We're now inside the abdominal cavity. Our mucosa has simple columnar epithelium, a little bit of lamina propria connective tissue, and then a thin layer of muscularis mucosa, smooth muscle. And then right below that is our submucosa, and then our muscularis externa. It's all smooth muscle at this point. There's no more skeletal muscle. Um, and this is where you find one of the differences in the stomach compared to the rest of the GI tract, is in the muscularis externa. We still have a circular layer and a longitudinal layer, but now we have an extra third layer that's inmost called the oblique layer. And then, of course, our serosa. So let's look at what that looks like. So here again, simple columnar epithelium in the mucosa. Um, so this is all mucosa. You can see the muscularis mucosa here underneath. We've got all our little gastric glands that are secreting mucus and pepsinogen for digesting proteins, and hydrochloric acid that makes our stomach acid. Below our mucosa is our submucosa, so that's this area here, and then our muscularis externa. And if we look at the muscularis externa in a little bit more detail, you can see the three layers in the muscularis externa. So now our inner layer is the oblique layer, that's this layer here. Then our circular layer, which is now the middle layer, and then the longitudinal layer is the last layer of the muscularis externa, and then our serosa again deep to that. The small intestine has three sections: the duodenum, the jejunum, and the ileum. They all have the same tunic structure. They all have the mucosa with the simple columnar epithelia. They all have areolar submucosal tissue. They have the bilayer muscularis externa, and again, areolar connective tissue in the serosa. They also have all of the same modifications to increase surface area. We have our plicae circularis, the folds in the mucosa and the submucosa that help slow food down as it travels through the small intestine. It helps increase surface area. And then our finger-like projections called the villi, and then the even smaller finger-like projections called the microvilli. So the amount of these surface area modifications decreases as we move towards the large intestine. So the duodenum, the first section of the small intestine, is going to have the most amount of villi, and the ileum will have the least amount of villi. And you'll see that when we look at the histology tissue sections. So the duodenum has lots and lots of villi. They tend to be kind of wide and quite tall. A distinguishing feature is the duodenal glands in the submucosa, and there are few goblet cells, not quite as many as there are someplace else's. The jejunum is our middle section, so the villi are becoming less numerous, but we still have lots of them. They're a little bit shorter. They're much narrower than they are in the duodenum, and we now do not have those duodenal glands in the submucosa. So that's a way that you could differentiate between the duodenum and the jejunum. The ileum is our last section, so we have the least amount of villi, because by at this point most of the absorption of our nutrients has really already occurred. We've got lots and lots and lots of goblet cells, and we have our lymphoid tissue clustered in those little Peyer's patches. So let's look at what these look like. Let's look at the duodenum first. We still have simple columnar epithelium lining each villus. You can see, so here's our mucosa. Lamina propria is inside, the kind of underneath, and then our muscularis mucosae. You can see the little crypts beneath the muscularis externa of the serosa, of the mucosa. We have our submucosa, so that's our next region. This is the submucosa of the duodenum. You can see here are our duodenal glands. You won't find these in other parts of the small intestine. And then we get our two muscle layers of the muscularis externa. We have the circular layer and then the longitudinal layer. And then finally we get our serosa. If we look at the jejunum, we still have lots of villi. The villi are a little bit shorter, a little bit more narrow. Again, you can see the lamina propria underneath all of the epithelia, and then the intestinal crypts, kind of the open spaces. Here's our little muscularis mucosae, so that little bit of smooth muscle that is part of the mucosa. Then we get our submucosa. You see, we no longer have those duodenal glands. They are not present in this part of the small intestine. They are only in the duodenum. And then our two bilayers, the circular layer of the muscularis externa and the longitudinal layer of the muscularis externa. And then here is where we would find our serosa tissue. If we look at the ileum, this particular section happens to be through one of the plicae circularis, so you can see kind of that fold. So this here is the mucosa. All this area here is mucosa. You can see we don't have as many villi. They're really narrow at this point, and there just aren't as many of them. All of this actually is submucosa, because the mucosa, of course, the plicae circularis are both folds in the mucosa and the submucosa. So all of this is submucosa, all of this here is submucosa, and you can see we've got these dark staining purple lymphoid nodules called Peyer's patches. And then our two layers of muscle in the muscularis externa, the circular layer and the longitudinal layer. And then the serosa would be below this. Looking at the large intestine, again, we've got our four tunics. We've got the mucosa with simple columnar epithelium, lamina propria, a little bit of muscularis mucosa. Got lots of goblet cells at this point. Then the submucosa of connective tissue, and then the muscularis externa. We have our circular layer, but one of the ways that the large intestine differs from the rest of the GI tract is the longitudinal layer is not a continuous band like it is elsewhere within the GI tract. It's been reduced to kind of three bands of smooth muscle that we call the tenei coli. And that creates those little like pouches, or those little haustra pockets in the large intestine. And then finally our serosa again. So if we look at our four layers, here is our mucosa. So all of this is mucosa. Then submucosa of connective tissue. And then here is our circular layer of the muscularis externa. And here is one of the tenei coli. You can see it's just this kind of little pocket of extra muscle, and it's absent here. And then over here again, we'll have another little pocket of little band of smooth muscle, and then our serosa tissue, connective tissue. At this point, it's mostly adipose tissue. As I said, the mucosa of the large intestine has lots and lots of goblet cells, and if we zoom in on the mucosa of the large intestine, you can see all of these little white spaces. This is a goblet cell, this is a goblet cell, this is a goblet cell. You got lots and lots of goblet cells lining the mucosa of the large intestine. If we look at our accessory organs, one of the first that food kind of comes in contact with is our salivary glands. We have two types of secretions. We have mucin, which is a lipid-based secretion, so mucus literally. So it tends to stain light pink or white. Anything that has lipid in it tends to kind of stain very lightly or even not at all. And then the other type of secretion is our serous fluid, and it's the serous fluid that actually contains our salivary amylase enzyme, which is a protein, and proteins tend to stain darker. They tend to light to dark pink sometimes, or even purple. Much, much darker than the mucin will stain. In terms of what these um, cells kind of look like, they're basically little clusters of cells, roughly triangular shaped, with the point of the triangle kind of pointing toward the duct. That way, as the secretion is made, it's kind of forced closer and closer towards the duct. Very often you will see that the serous cells kind of cap or wrap around the mucin cells as what we call a demilune, which literally means half moon. So these little half moon caps of serous cells around the clusters of mucin cells. Um, so we've got three salivary glands. We have the parotid, the sublingual, and the submandibular. The parotid secretes mainly serous fluid, although there are some mucus secreting cells. So since it secretes mainly serous fluid, it tends to be a darker tissue because it stains darker. The sublingual gland secretes mainly mucin, although again, there are some serous secreting cells. Since it secretes mainly mucin, it tends to have much more of a light color when we stain it because it's secreting mostly mucin, which is lipid-based, which tends to stain kind of light. And then our submandibular gland is our pretty evenly mixed gland that secretes both mucin and serous fluid, and so it will have a fairly good mix of both darkly staining serous cells and lightly staining mucin cells. And let's see what that looks like. So if we look at our parotid gland, you can see here are these kind of more darkly staining serous secreting cells. If we were to compare this darkly staining one of the parotid gland with the sublingual gland, you can see the sublingual gland is much, much lighter in color. So here is all of our kind of mucus secreting cells. But you can see we do have some serous demilunes, as you can kind of see here. But primarily what we have are our mucus secreting cells. If we look at the submandibular gland, you can see it's a pretty good mix of both serous cells themselves, serous cells in their demilune format, and mucus secreting cells. So the submandibular gland is a mixed gland that will have both the serous acini, the mucus acini cells, and the serous demilunes. If we look at the liver, you can see here your liver is a multi-lobed organ. So the inside each of those lobes of the liver are all of our liver lobules. So you're looking at a lobule here, a lobule here. Here's another lobule here. You can see the central veins, kind of right smack in the middle of the lobule, and then the portal triads where the arteriole and venule and bile duct tend to get kind of clustered at the corners of the lobules. If we look at one liver lobule in more detail, again, you can kind of here see our portal triad that has our arteriole, our venule, and our bile duct. There's another one here. Here's our central vein in the middle. And then you can kind of see these open spaces, and let's zoom in on that just a little bit further. So now we've zoomed in even further, and here's our central vein, and you can see all these clusters of cells are hepatocytes. Hepato meaning liver, cyte meaning cell, literally just means liver cells. So all of the darkly staining areas are our hepatocytes, our liver cells, and the more lightly staining areas are our sinusoids. Sinus typically means open space or fluid-filled space very often, and so they tend to stain light or white and not as dark pink. And then again, if we look here at our portal triad, you can see here's the venule, here's the little arteriole, and then here is the little bile duct. Then finally looking at another accessory organ, the pancreas. The pancreas has both an endocrine function and an exocrine function. So it functions as part of the endocrine system. That's the pancreatic islet. That's what's secreting insulin and glucagon directly into the bloodstream without any ducts. The islet here is our endocrine portion of the pancreas. And then the acinar portions of the pancreas, all of this is what is secreting all of our digestive enzymes. So it is the acinar portion of the pancreas that secretes the digestive enzymes into the ducts. They travel through the ducts into the small intestine. And so it is the acinar part of the pancreas that is part of our exocrine digestive system. So in terms of kind of what I want you guys to be able to do, you need to be able to look at a tissue and tell me what part of the GI tract does it come from. Is this from the esophagus? Is this from the small intestine? Is this from the large intestine? Is this the jejunum? Is it the ileum? Whatever the case may be. So you should be able to do that. You also need to be able to recognize our tunic structure. So be able to recognize the mucosa, the submucosa, the muscularis externa, and the adventitia or serosa. And then also you need to know how those tunics get modified for different parts of the GI tract. So whether it's the extra layer of muscle in the muscularis externa of the stomach, or the stratified squamous epithelium in the mucosa of the esophagus, whatever little modification gets made to the parts of the GI tract usually are a function of whatever they happen to be doing. So we've got stratified squamous epithelia in the mucosa of the esophagus because we've still got kind of whole food at that point, so it's a more protective function than the simple columnar where we've got lots of secretion and absorption going on in all the rest of the GI tract. We've got that extra layer of muscle in the stomach because that's where food really gets churned and pummeled and broken down physically. And not only do all of these little modifications relate to their function, but they're also some of the ways that you can look at a tissue slide and tell them apart. And then in terms of our accessory glands, you need to be able to identify the salivary gland. So look at a tissue slide, tell me, oh, this is from the parotid gland. Oh, this is from the sublingual gland. You need to be able to recognize the mucus cells and the serous cells, or the serous demilunes, as the case may be. And then also tell me what they secrete. So obviously serous cells secrete serous fluid, mucin cells secrete mucin. You need to be able to identify the different structures of the liver, so the lobule, the central vein, the sinusoids, the hepatocytes, the portal triad, whatever else was on that sheet that I gave you guys. And then in terms of the pancreas, be able to differentiate between the acinar portions and the islet portions of the pancreas. Tell me which portion is an exocrine function, so part of the digestive system, which part is the endocrine function, so part of the endocrine system, and what they secrete. So digestive enzymes coming from the acinar portions of the pancreas, and insulin and glucagon coming from the islets of the pancreas. So hopefully that helps clarify the histology a little bit.